Hi everyone, good evening. This is Dr. Mamta Jawa, your microbiology faculty. And in today's session, I have picked up my most favorite bacterium in the entire clinical bacteriology. Not because it's my most favorite, but also because it's the most favorite bacterium, probably of the examiners, also, as far as the clinically significant bacteria are concerned, because it's a very, very unique fellow, and because of its unique features, it is a favorite of the examiners, and you get a lot of questions on Listeria monocytogenes. So that was the reason behind choosing this very topic for this particular series, which we have started on the YouTube channel of PW MedEd. Okay, guys. All right. Listeria monocytogenes. So, guys, just like when we are born, our parents give us a nice name. Similarly, there's a basis behind the nomenclature of most of the microorganisms which we have. Like, for example, in this case, this fellow Listeria monocytogenes has been named after Sir Joseph Lister, regarded as the father of antiseptic surgery. All right. And hence the genesis of the word Listeria, the genus Listeria. And we have got the species name monocytogenes because this organism, this fellow, was found to cause multiplication of monocytes in rabbits, not in humans. Please remember, this feature was observed in rabbits and not in humans. This is very, very important to remember. Okay. Now, the most commonly used differential stain in any microbiology lab the world over is Gram stain. All right. Most important differential stain done is Gram stain. So, what is going to be the Gram reaction of this organism? Extremely important. This fellow is a Gram positive. Some books have mentioned it as rod, and some books have mentioned it as a cocobacillus. Now, cocobacillus means what? Rod is just like this perfectly cylindrical organism, this shaped. And cocobacillus means an organism which is neither perfectly spherical like a coccus, and nor it is a perfectly rectangular fellow like this rod. All right. So that is the meaning of cocobacillus. And now we'll see a beautiful gram stain picture of Listeria monocytogenes. See this. Okay, now this is a very, typical picture of the rod-like morphology of Listeria monocytogenes. But as I said, this fellow Listeria monocytogenes can also exist as, very, very short ovoid, that is oval forms, which are described as cocobacilli. So both the descriptive terminologies as far as the shape of this organism are concerned are correct. That is, it can be a rod and it can be a cocobacillus also. Okay. Now, why it is my favorite and why it is my most favorite and why it's the favorite of the examiners also, the reason is because of its unique features. Like, for instance, let me just point out some of the important unique features. This fellow is psychrotropic. This fellow is psychrotropic. Now, psychro means what? Psychro means low, and tropic means to have affinity for. Psychrotropic organisms are those which grow at both 37 degrees Celsius, obviously, because that is precisely why we are reading about them, because 37 degrees Celsius is the human body temperature, and these organisms are medically significant, whatever we read in clinical microbiology. So they obviously are supposed to grow at 37 degrees Celsius, otherwise how will they trouble us? So they are going to grow at 37 degrees Celsius, and Listeria monocytogenes in addition is going to grow at a lower temperature also. How low? It is going to grow at 4 degrees Celsius. So remember, L for Listeria, L for low, and how low the temperature can go, where at which it can survive, it can flourish well, it can multiply. The temperature is 4 degrees Celsius. Now let me know in the comment section where exactly do we have the temperature of 4 degree Celsius in our house? In our houses, throughout the 12 months of the year, we have got some place where the temperature is 4 degrees Celsius. Where is it? Very good, Sai Teja, fridge. So that means certain food items which are kept in the fridge, if suppose they happen to get infected or contaminated by this organism and they are consumed, then this organism can cause disease, can cause infection followed by disease in us. And obviously, please remember, infection does not mean disease. Okay, so in this context, please remember that Listeria monocytogenes is a saprophyte. Just like God, it is omnipresent. It's a saprophytic organism. It is a saprophyte and it is a food-borne pathogen. It is a food-borne pathogen. Just because somebody happens to ingest this organism along with the food items kept in the fridge doesn't mean that disease is definitely going to happen. Please remember, infection is not at all equivalent to disease. Okay, only when signs and symptoms, clinical signs and symptoms appear, then we say that disease development has happened. So it's a saprophyte and it's a food-borne pathogen. Then, so gram reaction, I have told you, the unique feature is that, another unique feature is that it is psychotropic, and yet another unique feature is that this fellow is despite being gram positive, this fellow is going to possess what? It is going to possess a deadly structure, namely endotoxin, also known as lipopolysaccharide. Because it is deadly, I am writing the name categorically in red color. And you all know that endotoxin or lipopolysaccharide generally is present where? It is generally present in the outer membrane, OMS outer membrane of G gram negative bacteria, GNBs, that is gram negative bacteria. It is an important integral structural component of the gram negative organisms, but Listeria monocytogenes is an exceptional gram positive organism which has got endotoxin or LPS in its structure. So that's another unique feature. Then what else? Now this fellow is motile, and the kind of motility which it exhibits is known as tumbling motility. Now what exactly is the meaning of tumbling? Tumbling means to fall suddenly. Like for example, I've got this pen with me. Now if suppose this falls suddenly like this is the literal meaning of tumbling. Now we are going to have a look at a video where we are going to appreciate the typical motility of this organism, that is the tumbling motility. See this. Okay, so on the screen, whatever moving things you are seeing, all of those are Listeria monocytogenes. So it is moving like this, falling suddenly. Looks like it is falling suddenly because of this precise appearance of its, this kind of motility. The designation of motility is tumbling motility. Okay. Then this fellow, okay, we have seen that it is motile, and the next important feature pertaining to motility which you need to remember is that it is motile. It is motile at 25 degrees Celsius, that is the environmental temperature, whereas it becomes NM, that is non-motile, at 37 degrees Celsius. Now I'll tell you both the real reasons and a memorizing tip to remember this. And by the way, before that, please remember what kind of behavior this is. This is referred to as what? This is referred to as differential motility. Differential motility. So remember, so far in this session, I have mentioned what? I have mentioned about differential stain, and now I'm mentioning about differential motility. That is, the motility is going to be, the behavior of the organism as far as motility is concerned is going to be different at two different temperatures. Okay. One second, guys. Okay. So we have got differential motility, we have got differential stain, and also we have got differential media also amongst the culture media of bacteria. Okay. So being motile at one temperature and non-motile at another temperature is referred to as differential motility. And whatever I've mentioned so far, guys, everything is an MCQ. Everything is an MCQ, be it your NEET PG exam, FMGE exam, INICET exam, and even if any second year student is watching this video, it's obviously important for your prof exam also. Okay. So so as I said, I'll tell you the real reason behind this behavior of differential motility, and I'll give you a memorizing tip also. Now the real reason is, see, whatever bacteria are motile, they are motile because of what? Flagella. And flagella express a protein called as flagellin protein. And being a protein, this protein is going to come from what? mRNA. mRNA is going to come from gene. All right. Now the gene for flagellin protein, the flagellar protein, is expressed at environmental temperature, and hence this organism is motile at 25 degrees Celsius, but this gene is not expressed at 37 degrees Celsius, and therefore this differential expression of the gene for the flagellar protein at two different temperatures is responsible for the differential motility of Listeria monocytogenes. Okay, I hope this is clear. And the memorizing tip which I would want to give all of you pertaining to this behavior is that I said that this organism, just like God, it is present, it is omnipresent, it is a saprophyte, it is there in nature. All right. So imagine this organism, while it is there in the surroundings, in the environment as a saprophyte, it is, it is on a mission. It is it has it has an aim, and the aim is to attack or target a human being. So in that quest of finding a human being whom it can infect and then cause disease, it is going to be motile in the environment. It will keep hopping from one place to another in the environment. And the moment it happens to infect a human being, that is the moment the temperature, the moment it encounters an area where the temperature is now 37 degrees Celsius, it will stop being motile because it would like to stay in the human body so that it can now, comfortably elaborate its valence factors and cause disease in this human being. Is this clear? Please let me know in the chat box if this is clear, or you have any doubts so far. You are more than welcome to ask. Any doubts, anyone? Okay. So it's a saprophyte, as I said, and we are studying it because it is medically significant. But please also remember that it infects not just humans, it infects a variety of animals also. Okay, it infects a variety of animals also. Okay, and then another unique feature of this organism is that it is facultative. It is facultative intracellular. That is, it has got the luxury or the blessing or the privilege to survive both outside the host cells as well as inside the host cells. And such organisms which are intracellular, be it obligate intracellular behavior or facultative intracellular behavior, they are notorious, most of them are notorious for inducing the formation of granuloma in the infected host tissues. And this also, you can take it as a reminder for another important disease, a very important disease caused by this organism, that is granulomatosis infantiseptica, which I'm going to elaborate upon just in a few minutes when we are going to learn about the clinical features. Okay, so ये facultative intracellular होता है. Then another important feature is that it is a facultative anaerobe. Facultative anaerobe. ठीक है. So facultative anaerobe का क्या मतलब है? It just means that it can survive both in the presence as well as in the absence of oxygen. So the word facultative actually means what? It means luxury or a blessing कि भाई दोनों चीजें हो सकती हैं. When we say facultative intracellular, we are trying to say that it can survive extracellularly and intracellularly also. So the faculty or the luxury or the blessing is to be intracellular. And when we say facultative anaerobe, we are trying to say that this organism is, per se its nature is that of an aerobic organism, but it has got the faculty or the luxury or the blessing to survive even in the absence of oxygen, so that is why it is called as facultative anaerobe. Okay, and then because it can grow at a low temperature, kitna low, 4 degrees Celsius. Okay, and that is why it's an important cause of gastroenteritis or food poisoning also, which again I'm going to elaborate upon just in a few minutes when we are going to learn about the clinical features. Also remember, because it can grow at such a low temperature, something known as cold enrichment can be done in the lab if at all you are contemplating to grow this organism on culture media. So cold enrichment can be done to grow it in the lab. Then how can we forget the two very very important enzymes when we learn about different bacteria, that is catalase and oxidase. So guys, this fellow is catalase positive and this is oxidase negative. ठीक है, and please remember these two features, catalase positivity and oxidase negativity. Can someone tell me in the comment section which important significant family or parivar of bacteria are defined by these two features? I mean, there are 10 important features basically of that significant family which has got so many pathogens and saprophytes also and commensals also. So these two are very very important features of those significant bacteria, but those are gram negative bacteria. Can someone tell me the name of that family? Anyone? Okay, I'll tell you. The name of that significant family is the family Enterobacteriaceae. Enterobacteriaceae, which is the family of gram-negative rods, which are catalase positive and oxidase negative. Okay? In the entire family of Enterobacteriaceae, there is only one catalase negative fellow, that is Shiga's bacillus or Shigella dysentery serotype 1. Okay? Okay, so this fellow is catalase positive, oxidase negative, and also remember this fellow is weakly beta hemolytic. When I say hemolysis, obviously I'm gonna use what medium I'm to comment upon hemolysis, blood agar. So it's weakly beta hemolytic, and this beta hemolysis is seen because of production of Listeriolysin O. Accepted short form of Listeriolysin O is LLO. This is not my own short form. This is the accepted short form, and this is an important VF, that is virulence factor also. That is one of the important arms and ammunitions or अस्त्र और शस्त्र of this organism. Okay, so चलो, talking about virulence factors, एक तो मैंने बताई दिया, one is Listeriolysin O, and remember another important virulence factors are phospholipase enzymes. Okay, phospholipases. ठीक है, एंड येट अनदर यूनिक फीचर. अभी तक मैंने जो बोला है, वट एवर आई हैव मेंशन अबाउट दिस ऑर्गेनिज्म सो फार, एवरीथिंग इज यूनिक ओनली. इट इज साइक्रोट्रोपिक, इसके पास एंडोटॉक्सिन होता है. मॉर्फोलॉजी वाइज भी ये कंफ्यूज हमें कर सकता है, बिकॉज इट कैन बी अ कोकोबैसिलस और इट कैन बी अ रॉड. Okay, then differential motility I have told you. The special name of motility is tumbling motility. Then another unique feature is that it jumps from one cell to another via formation of what? Via formation of what is known as actin rockets. Actin rockets. Now we are going to learn how exactly these actin rockets are formed and what they are basically. Now have a look at this. Okay guys, let me zoom the picture. Now at the periphery of this, and on your screen you are seeing in the center, schematic representation is there where Listeria is going to invade a cell, and we are going to learn in just a few minutes how it hops onto the adjacent cell, and at the periphery you can see the electron microscopic picture of the same thing happening. Okay, so it's easier to learn this with the help of the schematic representation, then you can closely correlate it with the electron microscopic picture as well. So we have got INLA and INLB. What are these? These are internalins. These are Internalins. What are internalins? These are receptors on the surface of the host cell. Let's say this is host cell number 1 and this is host cell number 2. And this fellow, the blackish fellow, this is Listeria. Let me label it L. L for Listeria. So what happens? Listeria attaches to the host cell receptors, namely internalin A, or it could be internalin B. So these receptors, as their name implies, they're going to internalize Listeria, and Listeria, congratulations, now it has entered inside this host cell in what? In this sac, namely phagosome. Okay, phagosome. But remember the two important valence factors which I mentioned, listerio-lysin O and phospholipases. It will elaborate these valence factors and it will break down the membrane of the phagosome, as you can see over here. Let me star marked it. And so now we have got Listeria monocytogenes free in the cytoplasm. Now it is lying freely in the cytoplasm. So once it is free in the cytoplasm, it is going to do a very, very important activity, and that is the decoration or flaunting or showing off of some protein on its surface. What is the name of that protein? The name of that protein is Act A. Okay, the name of that protein is Act A. Now Act A, as the name implies, it has got to, it is, it is going to be a very active protein, in the sense that it is going to bring about some important activity. And what is that important activity? That important activity is the polymerization of actin filaments which are there in the cytoplasm of this host cell. So as you can see, these, these grayish color things, as if, something has been scribbled around this dark yellow, this dark black organism. So what is this? This is nothing but the polymerization of the actin filaments, and it is behind this bacterium, so it is referred to as actin tail. And with the help of this, now a projection of the cell membrane is going to be produced like this, and subsequently it is going to be, pushed or injected into the adjacent cell, just like, we are injecting some drug with the help of a needle and syringe, or just like a slingshot. ठीक है, तो इस तरह से now this organism in this sac, it is going to be entering this host cell number 2 again in the phagosome, which again will be broken down by LLO and phospholipases, and again it is going to lie free in the cytoplasm, and again it will do the same thing. It will proudly flaunt or decorate or show off what? Act A protein, which is going to cause polymerization of the actin cytoskeletal filaments in the host cell. So what is the advantage of all of this? What is the, why God has blessed this particular bacterium with all of this? What is what this organism is going to evade with this behavior? Can someone tell me? Two important aspects of immune system, two important types of immunity are what? Humoral immunity and cell mediated immunity. Can someone comment in the chat box and let me know what kind of immunity is being evaded by this fellow because of this very nature of hopping on from one or jumping from one cell to another? Batao. Because of this very behavior of jumping from one cell to another via actin rockets, this, that is why this fellow is what? Facultative intracellular, cell के बाहर भी रह सकता है, अंदर भी रह सकता है. And which kind of immunity is being evaded? It evades what? It is going to evade antibodies, that is humoral immunity. It evades humoral IR, that is immune response. It evades humoral immune response. ठीक है ना? So this obviously is going to give an edge of this organism. So guys, please remember, microbiology is not at all about rote learning. There's a reason or logic behind most of the things. रट्टा तो आपको हर सब्जेक्ट में ही मारना है, किसमें नहीं मारना है, 19 सब्जेक्ट्स में ही मारना तो पड़ता ही है. So facts you have to remember, but unlike the popular belief that it is all about, micro is all about rote learning, please, it is not. It is so not. ठीक है? I hope this is clear, everyone. So this explains the facultative intracellular nature of this organism, and the very fact that what branch of immune system is going to be significant pertaining to this organism. CMI is more important. Cell mediated immunity is more important, way more important against Listeria monocytogenes than humoral immunity. Okay. Now other bacteria. So this fellow is very, very smart, isn't it? It can just hop on from one cell to another via these actin rockets. Now there are other bacteria also which use this strategy, and therefore they can also evade the antibodies. And who are these bacteria? RMSB. Now R stands for what? Rickettsia. M stands for Mycobacterium. S stands for Shigella, and B stands for Burkholderia. Burkholderia. Okay. And the next important thing which you need to know, this again, this is an old MCQ, but they can still ask about the serotypes, the medically significant serotypes of Listeria monocytogenes. Okay. So the important serotypes which happen to infect and cause disease in humans include 1 by 2A, 1 by 2B and 4. Okay. So please remember this. Okay. Now important tests to identify, I mean important tests, not exactly to identify, because for identification we have to do a lot more other tests to arrive at the conclusion that it is definitely Listeria monocytogenes. Some important tests which are still done as a tentative, they can give us a tentative idea ki acha this could be Listeria. One is the CAMP test, stands for Christie Atkins Munch Peterson. These are not four people, these were actually three people who devised this test first in the context of whom? Group B, Streptococcus or Streptococcus agalactiae. This test is a test of synergistic hemolysis. Synergistic hemolysis. Okay. Now we have learned that this fellow Listeria monocytogenes kya hai? It's weakly beta hemolytic because of listeriolysin O. All right. So in CAMP test, we show the synergistic hemolysis by Staph aureus and Listeria monocytogenes. Originally, this was devised in the context of GBS by these people, Christie Atkins Munch Peterson. Okay. Then the next test, which is not done nowadays because of ethical reasons, but MCQs are commonly asked on this, is Anton's test. Now this is very similar to whom? This is very similar to the Serenis test. Serenis test किसके लिए करते हैं? Serenis test अभी नहीं करते, I mean करते थे पहले. So basically Serenis test was done earlier on for entro-invasive E. coli. This is a test of invasiveness. ठीक है, तो करते हैं क्या है इस केस में? Anton's test या Serenis test. अगर Serenis, so basically you are taking an animal and you are going to inject a filtrate of prepared from the colonies of the organism into the lower conjunctival sac of the animal, and you will see whether keratoconjunctivitis happens or not. What you are trying to show by this, you are trying to show the invasive property of the bacteria. If at all you are suspecting, you have grown colonies of entro-invasive E. coli, you have got strong suspicion that it is EIEC only, then you are going to call it Serenis test. Or if suppose you have concluded that whatever organism you are dealing, whatever organism your patient is infected with is Listeria monocytogenes, and you want to carry out this test of invasiveness in the animals, then you're going to call it Anton's test. Now let me show you a picture. You are not going to find this picture on Google. It's a very rare pic. See this. चलो, इन दिस पिक्चर वी आर सीइंग अ गिनी पिग, लेकिन रैबिट भी ले सकते हैं या ले सकते थे, अभी तो नहीं करते हैं आजकल ये चीज़ें. सो गिनी पिग या रैबिट हमने लिया, ठीक है, एंड व्हाट हैपेंड टू द पुअर फेलो, इट डेवलप्ड केरेटोकंजंक्टिवाइटिस. क्यों? आफ्टर कंजंक्टिवल इनोकुलेशन ऑफ इन्वेसिव एस्चेरिशिया कोलाई. सो दिस पिक्चर प्रिसाइजली इज दैट ऑफ the test of invasiveness being done for entro-invasive E. coli. So ये Serenis test है. This is a positive Serenis test. So as I said, no longer used in diagnosis. ठीक है. So इसी तरह से अगर suppose you probably are thinking in terms of on the lines of Listeria monocytogenes. आपने ग्राम स्टेन कर लिया, आपने देख लिया कि इट्स अ ग्राम पॉजिटिव फेलो, टम्बलिंग मोटिलिटी भी दिख गई, कॉलोनीज कुछ आ गई, वीकली बीटा हेमोलिटिक कॉलोनीज मिल गई आपको, तब आप अगर ये टेस्ट करते हो तो आप इसका नाम क्या दोगे? देन यू आर गोइंग टू कॉल इट एंटन्स टेस्ट. ठीक है, देन यू आर गोइंग टू कॉल इट एंटन्स टेस्ट. Okay, now the diseases by Listeria monocytogenes. One, of course, is gastroenteritis, as I mentioned earlier on when I was elaborating about the psychotropic nature of this organism. Now, very, very importantly, incubation period is said to be more than 16 to 18 hours. Sometimes it could be 2 days, that is 48 hours, and this is mentioned only and only in Harrison and nowhere else. And I've seen an MCQ, a clinical vignette on bacterial food poisoning, where they mentioned the food item and the incubation period was mentioned as 2 days. ठीक है, तो इंक्यूबेशन पीरियड थोड़ा सा इधर उधर हो सकता है. तो टू आंसर क्वेश्चंस ऑन बैक्टीरियल फूड पॉइजनिंग, विच बाय द वे इन इट सेल्फ इज अ वेरी वेरी इंपॉर्टेंट टॉपिक, यू हैव टू नो वन द क्लिनिकल फीचर्स की वॉमिटिंग है, डायरिया है, डिसेंट्री है. नंबर 2 इंक्यूबेशन पीरियड. नंबर 3 व्हाट फूड आइटम्स वर कंज्यूम्ड बाय द पेशेंट. ठीक है, तो अगर ये तीनों चीज़ें पता है तो ईजिली यू विल बी एबल टू सॉल्व दीज क्वेश्चंस. ठीक है, सो ऑन पर्पज आई एम मेंशनिंग दिस 48 आवर्स ड्यूरेशन ऑल्सो एज इंक्यूबेशन पीरियड, क्योंकि ये सिर्फ हैरिसन में है और क्वेश्चन आ चुका है इसके ऊपर. ठीक है, नाउ सो इंक्यूबेशन पीरियड एज ए सेट पता होना चाहिए, फूड आइटम्स पता होने चाहिए. कौन कौन से फूड आइटम्स इम्प्लीकेटेड हैं? नाउ बहुत सारे हैं एक्चुअली, लेकिन मोस्ट इंपॉर्टेंटली जिन पे क्वेश्चन आता है ना, दे आर मिल्क एंड मिल्क प्रोडक्ट्स, स्पेशली सॉफ्ट चीज. स्पेशली कौन सा? सॉफ्ट चीज. ठीक है, अब हैव अ लुक एट दिस. चलो, नाउ लेट अस सी. मैंने काफ़ी टाइम लगाकर इस चीज़ को बनाया था. आई होप इट हेल्प्स यू गिव अ विजुअल इम्प्रेशन. ठीक है, सो दिस इज अ क्यूट गर्ल हु इज प्रेइंग टू द लॉर्ड वेरी वेरी सिंसियरली. ओ डियर लॉर्ड, प्लीज फुलफिल माई विश लिस्ट. लिस्ट को मैंने जानबूझकर ब्लू कलर में लिखा है जस्ट टू गिव यू मेमोराइजिंग टिप कि भाई लिस्टीरिया क्या है, ग्राम पॉजिटिव है, और ये प्रे कर रही है बहुत सिंसियरली, बहुत आर्डेंटली भगवान से कि इसकी विशेस पूरी हो जाएं. तो इंक्यूबेशन पीरियड रिमेंबर 48 आवर्स एंड तो इसकी so she's ardently praying to the Lord, and you can see she's in not some religious place, and she in front of her you can see different food items which you have to remember in the context of listeria food poisoning: soft cheese, delicacy meat, milk and hot dog. ठीक है, एज आई सेड क्वेश्चंस मोस्टली सॉफ्ट चीज़ पे आते हैं. मिल्क एंड मिल्क प्रोडक्ट्स आपको नहीं भूलना है बिल्कुल भी. सो रिमेंबर द फर्स्ट एंड फॉरमोस्ट डिजीज विच यू नीड टू रिमेंबर पर्टेनिंग टू लिस्टीरिया मोनोसाइटोजीन्स इज गैस्ट्रोएंटेराइटिस. ठीक है, इंक्यूबेशन पीरियड मत भूलना, 48 आवर्स याद करना, वेरी वेरी इंपॉर्टेंट. एंड मोर देन 16 to 18 आवर्स वैसे होता है. इंक्यूबेशन पीरियड कोई पत्थर की लकीर नहीं है, थोड़ा बहुत दे सकते हैं. ठीक है, तो द क्लिंचर टू डायग्नोसिस ऑफ फूड पॉइजनिंग केसेस बाय बैक्टीरिया आर, एक तो क्लिनिकल फीचर्स क्या हैं, और दूसरी चीज फूड आइटम्स कौन से कंज्यूम किए थे पेशेंट ने. ओके, सो क्लिनिकल फीचर्स. तो क्लिनिकल फीचर्स क्या हो जाएंगे? फीवर, हेडेक, डायरिया एंड कॉन्स्टिट्यूशनल सिम्टम्स. अच्छी बात क्या है इस फूड पॉइजनिंग के बारे में? द गुड थिंग इज दैट दिस फूड poisoning is a self-limiting condition. You need not give any antibiotic to the patient. ठीक है, और बैक्टीरिया होता है, बैक्टीरिया अकर्स, लेकिन वो भी डेंजरस नहीं है बिकॉज इट इज एज आई सेड सेल्फ लिमिटिंग कंडीशन. इम्यून सिस्टम इज गोइंग टू टेक केयर ऑफ बैक्टीरिया विच इज गोइंग टू बी ओनली ट्रांसिएंट इन केस ऑफ लिस्टीरिया गैस्ट्रोएंटेराइटिस. ठीक है, सो दैट्स अबाउट द वेरी फर्स्ट डिजीज. उसके बाद आते हैं सेकेंड डिजीज पर, दैट इज सेप्टिसीमिया. सेप्टिसीमिया बाय लिस्टीरिया मोनोसाइटोजीन्स. इसमें पेशेंट को क्या होगा? दिस इज नाउ गाइज अ सीरियस थिंग. दिस इज द पेशेंट इज गोइंग टू हैव फीवर, chills, myalgia, arthralgia. ठीक है, एंड ऑल्सो रिमेंबर 70 से 90%, 90-90%, 70 to 90% ऑफ कैंसर पेशेंट्स, 70 to 90% ऑफ कैंसर पेशेंट्स with listeriosis. Listeriosis is the designation for infection with Listeria monocytogenes. 70 to 90% of cancer patients with listeriosis have bacteremia. Have bacteremia. ठीक है. एंड देन बिकॉज बैक्टीरिया करा रहा है, सेप्टिसीमिया करा रहा है, तो एंडोकार्डाइटिस भी कुछ लोगों को हो सकता है एंड इट कैन इन्वॉल्व बोथ प्रोस्थेटिक हार्ट वाल्व्स एज वेल एज द नेटिव वाल्व्स. ठीक है. देन तीसरी चीज, तो वी आर गोइंग फ्रॉम लेस सीवियर टू मोर सीवियर थिंग्स. फूड पॉइजनिंग उतनी प्रॉब्लमैटिक नहीं है अगर तो इम्यूनिटी अच्छी है. देन प्रॉब्लमैटिक क्या हो सकता है? सेप्टिसीमिया हो सकता है. देन थर्डली प्रॉब्लमैटिक चीज़ क्या ये करा सकता है? बैक्टीरिया ड्यूरिंग प्रेगनेंसी, बिकॉज रिमेंबर प्रेगनेंसी अपने आप में एक इम्यूनो कॉम्प्रोमाइज्ड स्टेट ही है. ठीक है. एंड स्पेशली इफ बैक्टीरिया अकर्स, इफ इट अकर्स इन थर्ड ट्राइमेस्टर में इन्फेक्शन हो गया, एक प्रेग्नेंट लेडी को बैक्टीरिया हो गया, तो क्या होगा? द बैक्टीरिया कैन सीड द प्लेसेंटा, दे कैन सीड द प्लेसेंटा, therefore the bacteria can cross, they can cross placenta also, and this can have disastrous consequences unfortunately, जैसे कि abortion या still birth हो सकता है, या फिर प्री टर्म लेबर हो सकती है, कोरियो एमनियोनाइटिस हो सकता है. ऑलराइट. और प्रेग्नेंट लेडी को फीचर्स, क्लिनिकल फीचर्स क्या होंगे? द लेडी विल कंप्लेन ऑफ फीवर, एफ फॉर फीवर, देन बैकएक, myalgia. So this can have disastrous consequences if if infection occurs in the third trimester. उसके बाद fourthly, infection can even occur near term. Okay, so if infection in a pregnant lady occurs, if infection occurs towards term, okay, the baby in utero is going to get infected and the baby can develop, as I mentioned before also, I mentioned something in the context of in the context of the facultative intracellular nature of this organism कि भाई these organisms can cause granuloma in the infected host tissues. तो बेबी को अब क्या बीमारी हो सकती है, क्या डिजीज हो सकता है? ग्रैनुलोमेटोसिस इन्फैंटी सेप्टिका. ठीक है. ग्रैनुलोमेटोसिस इन्फैंटी सेप्टिका, एंड दिस एंटिटी इज कैरेक्टराइज्ड बाय व्हाट? It is characterized by granulomas, granuloma formation in liver, spleen and adrenals. ठीक है. उसके बाद now there is something called as neonatal listeriosis, which most commonly presents as meningitis, and it can even present as septicemia. So Listeria monocytogenes is an important cause of neonatal meningitis and neonatal septicemia. ठीक है. Now I'll make a box and mention the organisms, or let me first show you. Okay, let me just show you the picture right away. Now see this. All right. So important causes of neonatal meningitis can easily be remembered by remembering this this picture of this cute kid and remembering this sentence from top to bottom. Play with the leg of the neonate. I have already long back I pasted this picture on my Instagram handle also, mj.micro. ठीक है. सो प्ले विद लेग ऑफ द नियोनेट. तो एल से क्या है? एल से लिस्टीरिया मोनोसाइटोजीन्स. On purpose, I have written the name in blue color to help you remember that this organism is a gram positive organism. फिर enterobacteriaceae मैंने कहीं पे mention किया आज. enterobacteriaceae, yes I did, when I told you that this organism Listeria monocytogenes is catalase positive, oxidase negative, and I happen to tell you that same two features are also there in a very significant परिवार और family of bacteria of gram negative rods, that is enterobacteriaceae. So gram negative है तो इसलिए मैंने नाम red में लिखा है. And in among so many members of enterobacteriaceae, neonatal meningitis कौन कराता है? एस्चेरिशिया कोलाई एंड क्लेप्शियला निमोनिया. उसके बाद जी से क्या है? ग्रुप बी स्ट्रेप्टोकोकस. ग्रुप बी स्ट्रेप्टोकोकस या जीबीएस या स्ट्रेप्ट ए गैलेक्शिया मैंने कहाँ मेंशन किया है आज? मैंने आज मेंशन किया था कैम टेस्ट के कंटेक्स्ट में. सो गाइज प्लीज रिमेंबर, इसी तरह से ना आपको पढ़ना भी होता है. इफ यू आर रीडिंग सम टॉपिक एंड यू हैपन टू हैव सम डेजा व्यू अबाउट सम वर्ड, सम फ्रेज कि भाई ये तो थोड़ी देर पहले मैंने पढ़ा था या कहीं तो पढ़ा था, तो आपकी रिस्पॉन्सिबिलिटी क्या होती है कि आपको उसी टाइम कायदे से देखना है कि वेयर यू रेड इट, flip the pages, it might take just, just a few minutes. It's not going to take long. And I would say, even if it is taking you long, a little bit long, that's okay, because this is how you are actually going to make picture memory. और एग्जाम में पिक्चर मेमोरी ही काम आती है, लॉन्ग टर्म मेमोरी काम आती है, ओके, मेमोरी सेल्स काम आएंगे, ठीक है. बिकॉज इफ टुमारो इज योर एग्जाम, नोबडी कैन रीड, नोबडी ऑन मदर अर्थ कैन रिवाइज ईच एंड एवरी वर्ड ऑफ द नोट्स और द मटेरियल विच यू हैव. सो दिस इज वन गुड वे ऑफ मेकिंग लॉन्ग टर्म मेमोरी, कि अगर आप कुछ भूल गए हो तो उसी टाइम चेक करो कि अच्छा ये कहाँ पढ़ा था, और उसी टाइम करना है, बिकॉज बाद कभी नहीं आता है. ओके, okay, so it causes meningitis in neonates. It's an important cause of neonatal meningitis. उसके अलावा it also causes meningitis in I/C people, that is immunocompromised people, especially after क्या, especially after organ transplantation. Then it also causes meningitis in geriatric population, that is again. So हमने क्या देखा, एक्सट्रीम्स ऑफ एज में करता है मेनिंजाइटिस, न्यूनेट्स में कर रहा है और गेरियाट्रिक पॉपुलेशन में कर रहा है. लेकिन प्लीज रिमेंबर, अगर गेरियाट्रिक पॉपुलेशन की बात करें, इन दिस कैटेगरी और स्ट्राटा ऑफ इंडिविजुअल्स, दैट इज गेरियाट्रिक पॉपुलेशन, द एमसीसी, मोस्ट कॉमन कॉज ऑफ मेनिंजाइटिस कौन होते हैं? द एमसीसी या मोस्ट कॉमन कॉज ऑफ मेनिंजाइटिस आर ग्राम नेगेटिव एंटेरिक रॉड्स. ठीक है, ग्राम नेगेटिव एंटेरिक रॉड्स. ऑलराइट, देन there's also something known as listeriosis, not associated with pregnancy. Some other significant conditions, other than pregnancy, where we saw it can have catastrophic consequences like abortion वगैरह वगैरह. So some of the other special conditions where listeria infection can be really dangerous include: number 1, chronic glucocorticoid therapy. ठीक है, उसके बाद नंबर 2 इज डायबिटीज मेलाइटिस. देन थर्ड वन इज सॉलिड एंड हेमाटोलॉजिक malignancies, especially, now this is very very important, especially fludarabine treated, where treatment has been given with the fludarabine, especially those malignancies. ठीक है, now अब हम आते हैं diagnosis of listeriosis. Now see this, okay, before diagnosis, now also have a look at this. Acha, so what I have drawn over here, I have drawn a pregnant lady. ठीक है, you can see the baby bump, and the lady is having a craving to have ice cream. I have loads of ice cream, in other words listeria, because this ice cream has been contaminated with listeria, but obviously we can't see the bacteria with naked eyes, so she's just going to relish the ice cream and enjoy it. But तो यहाँ पे जैसे मैंने जूम करके दिखाया हुआ है. सो लिस्टीरिया, प्लीज रिमेंबर अगेन, दिस इज काइंड ऑफ अ रिविजन फॉर द इंपॉर्टेंट फीचर्स फॉर यू. लिस्टीरिया मोनोसाइटोजेंस क्या देख रहे हो आप? दिस इज अ ग्राम पॉजिटिव रॉड या कोकोबैसिलस. अरेंजमेंट ऑफ फ्लैजिला क्या है? पेरीट्राइगस फ्लैजिला. पेरी मीन्स व्हाट? पेरीमीटर मतलब ऑल अराउंड. पेरीट्राइगस फ्लैजिला से ये मोटाइल होता है. रिमेंबर, मोटाइल एट 25 डिग्री सेल्सियस एनवायरमेंट में, बट नॉन मोटाइल एट 37 डिग्री सेल्सियस. ग्राम पॉजिटिव है, लेकिन इट हैज गॉट एंडोटॉक्सिन. रेड में इसलिए लिखा है बिकॉज एंडोटॉक्सिन अदरवाइज कहाँ पे होता है? ग्राम नेगेटिव फेलोज में होता है, जो रेड दिखते हैं ऑन ग्राम स्टेन. ठीक है, सो दीज आर सम ऑफ द इंपॉर्टेंट फीचर्स यू नीड टू रिमेंबर. अब आगे देखते हैं डायग्नोसिस के बारे में. ओके, सो दीज आर द सैम्पल्स यू गोइंग टू कलेक्ट. ब्लड सैम्पल ले सकते हो, एमनियोटिक फ्लूइड, सेरेब्रोस्पाइनल फ्लूइड. ठीक है, देन ग्राम स्टेन करोगे तो यू विल सी व्हाट यू हैव टू डिस्क्राइब. ग्राम स्टेन पे हाउ डू यू कमेंट? यू हैव टू कमेंट ऑन द ग्राम रिएक्शन, ग्राम पॉजिटिव है या नेगेटिव है, मॉर्फोलॉजी. तो इस पिक्चर में दिस आई शोड यू अर्लियर ऑल्सो. सो यहाँ पे यू कैन कमेंट दैट यू आर गोइंग टू यू आर सीइंग ग्राम पॉजिटिव रॉड्स. दैट्स ऑल, नो स्पेशल अरेंजमेंट इज देयर. ठीक है, नाउ उसके अलावा, नाउ सी दिस, देन ब्लड अगार इज अ वेरी कॉमनली यूज एनरिच्ड मीडिया इन सो मेनी लैब्स. तो आप सैंपल उस पर इनोकुलेट करोगे एंड इन केस ऑफ लिस्टीरिया यू विल सी बीटा हीमोलाइसिस. इट इज वीक बीटा हीमोलाइसिस एट्रीब्यूटेड टू व्हाट, लिस्टीरियो लाइसिन ओ और एलएलओ. ठीक है. उसके अलावा नाउ सेलेक्टिव मीडियम फॉर दिस ऑर्गेनिज्म क्या है? द सेलेक्टिव मीडियम इज पैल्कम अगार. सेलेक्टिव मीडियम से क्या मतलब हुआ? सेलेक्टिव, देखो पार्शियलिटी सिर्फ लाइफ में नहीं होती हम लोगों के साथ, पार्शियलिटी ये मीडिया भी करते हैं. सेलेक्टिव मीडिया मीनिंग दीज मीडिया आर इनकॉर्पोरेटेड विद सर्टन सब्सटेंसेज which are going to show selectivity or partiality or favoritism towards the growth of only specific microorganisms and not others. So when I say selective medium for Listeria monocytogenes, I am referring to a medium which is going to allow the growth of only Listeria monocytogenes if at all it is there in this clinical specimen जो आपने उस पर inoculate किया है. If at all other organisms are there in the specimen of the patient, they will not grow. Listeria है तो करेगा ग्रो, और बाकी ऑर्गेनिजम्स अगर तो होंगे तो वो ग्रो नहीं करेंगे. सो सेलेक्टिव मीडियम इज पैल्कम अगार वेयर पी स्टैंड्स फॉर पॉलीमिक्सिन. ठीक है. ए स्टैंड्स फॉर एक्रीफ्लेविन, एल स्टैंड्स फॉर लिथियम क्लोराइड. ठीक है. सी स्टैंड्स फॉर सेफ्टाजिडिम, ए स्टैंड्स फॉर एस्क्यूलिन, और एम से क्या है? मैनिटॉल. पैल्कम अगार, अगेन ये क्वेश्चन है इस सेशन में या कोई भी वीडियो लेक्चर्स जो करे हैं मैंने पीडब्ल्यू मेडेड ऐप पर, या जो लाइव क्लासेस भी चल रही हैं, ऐसा कुछ चीज़ नहीं है जो एग्जाम में नहीं आता है या एज मेडिकल्स आपको नहीं पता होना चाहिए. सब कुछ इस पर इज वेरी वेरी रेलिवेंट टू योर एग्जाम्स. ठीक है. तो कैम टेस्ट कर सकते हो, कैम टेस्ट इज पॉजिटिव एज आई सेड, एंड कैटलेस टेस्ट कर सकते हो, दिस फेलो इज कैटलेस पॉजिटिव. ठीक है. एंड remember again it's a facultative intracellular organism. ठीक है. This you need to remember in the context of culture of this organism. You need not have any anaerobic conditions. तो facultative intracellular है. ठीक है. facultative anaerobic है, तो that you have to remember. एंड एज आई सेड कि कोल्ड एनरिचमेंट कर सकते हैं कल्चर के लिए. तो उसके कंटेक्स्ट में एक चीज़ और याद करो कि देयर इज समथिंग नोन एज ट्रिप्टिकेस सोय ब्रॉथ. ठीक है. यू ऐड द सैंपल इन ट्रिप्टिकेस सोय ब्रॉथ एंड यू इंक्यूबेट इट एट 4 डिग्री सेल्सियस. ठीक है. And this is going to be followed by F/B is not Facebook, it is followed by subculture onto the plates. कौन सी प्लेट? सेलेक्टिव मीडिया. एक सेलेक्टिव मीडिया मैंने आपको बता दिया पैल्कम अगार, बिकॉज आपको अगर आइडेंटिफाई करना ऑर्गेनिज्म दैट यू कैन डू बाय गेटिंग प्योर कॉलोनीज विच यू आर गोइंग टू ऑब्टेन ओनली ऑन सेलेक्टिव मीडिया. सेलेक्टिव मीडिया हमेशा सॉलिड होते हैं. एस फॉर सेलेक्टिव, एस फॉर सॉलिड. ठीक है. तो ट्रिप्टिकेस सोय ब्रॉथ तो लिक्विड मीडियम हो गया. तो पहले सैंपल को इसमें डालो, उसको अच्छे से अलाउ करो ग्रो करने के लिए बाय इंक्यूबेटिंग एट 4 डिग्री सेल्सियस. जब वो ग्रो कर जाए देन यू हैव टू सबकल्चर ऑन टू सेलेक्टिव मीडिया. इज दिस क्लियर एवरीवन? ओके. नाउ लास्टली द ट्रीटमेंट एस्पेक्ट. नाउ a kind of antibiotics which are blatantly used unfortunately in India, because यहाँ पे तो over the counter सब कुछ मिल जाता है. they are cephalosporins. इतनी blatantly यूज होती हैं, कई बार बहुत बार मिसयूज भी होती हैं, because they are one of the safest antimicrobials to be used. and तो क्या होता है कि अगर आप लिस्टीरिया लेकिन अगर सस्पेक्ट कर रहे हो तो यू हैव टू कीप इन माइंड, यू हैव टू बी रियली माइंडफुल ऑफ द फैक्ट दैट cephalosporins supposed to be of value in a lot of infections, in a plethora of infections, broad range of infections. They are going to be a failure in case of listeria. So please remember, treatment के लिए the rule which you have to remember is that cephalosporins are ineffective. They are not effective अगर आप listeria suspect कर रहे हो. I am emphasizing upon this so much क्योंकि imagine करो ये ऑर्गेनिज्म जो हमने देखा इतने डेडली डिजीज कर सकता है. लाइक फॉर एग्जांपल प्रेग्नेंट लेडी है, यू कैन नॉट रिस्क द लाइफ ऑफ द मदर एंड द बेबी बाय गोइंग अहेड विद सेफलोस्पोरिन. सो अगर आपको लिस्टीरिया हैज़ टू बी एज वन ऑफ योर डीडीज. ठीक है. तो उस केस में आप सेफलोस्पोरिन प्लीज़ मत यूज़ करो, अदरवाइज यू माइट लूज द पेशेंट. तो ट्रीटमेंट के लिए द ड्रग ऑफ चॉइस क्या है? यूजुअली वी गिव अ कॉम्बिनेशन ऑफ एम्पिसिलिन प्लस जेंटामाइसिन, एम्पी जेंटा कॉम्बिनेशन। कई बार क्या होता है, माइल्ड डिजीज के लिए सम क्लिनिशियंस प्रेफर ओनली एम्पिसिलिन, बट फॉर सीवियर डिजीजेस ऑब्वियसली लाइक फॉर meningitis and all you have to go for ampygenta combination. And in case of penicillin allergy, in case of penicillin allergy you can use cotrimoxazole. ठीक है, you can use cotrimoxazole. I'm absolutely fine, Sunil. What happened? I just noticed this message. Okay, so as I said, the drug of choice क्या होगी इसकी? Ampicillin plus gentamicin. Okay, guys. So that's about today's session. I thank all of you for your time and patient listening. Okay, if you have any doubts, you can mention in the comment section. All right, you can follow my micro handle where I'm most active, mj.micro, the Instagram handle, and check out the description of this video where you can find out, all the social media handles of PW MedEd. You can join the Telegram group also. The Facebook group also is there, and PW MedEd handle is there on Instagram also. So stay connected with us, and in case you are stuck anywhere in regard to the with regard to the use of the app, any problem with subscription or anything, just let us know, and the team members will definitely sort it out. Any doubts, anyone? This topic, by the way, is very important. So I guess there are no doubts. Hence, I'm going to stop streaming right now. Okay, once again, thank you everyone.